Welcome to Biology at Fujit, my dear students. Today we explore an important concept that changed our understanding of heredity, that is, the fine structure of the gene. A gene is not a single individual unit, but it is basically a finely organized sequence of DNA with distinct sites for function, mutation, and recombination. Through classic experiments, scientists like Seymour Benzer revealed that genes have an internal architecture allowing precise mapping within a single gene itself. The deeper insight helps us understand how genetic variations arise and how traits are accurately controlled at the molecular level. Let's take a closer look at this fascinating micro world hidden within a gene. History. The term gene was firstly introduced by Johansen in 1909 based on Mendelian factors. Gene concept was given by Sutton and Boveri. Further studied and it was elaborated by Morgan, Bridges and Muller. So we go through the summary of evolution of the gene concept. In 1866, Gregor Johann Mendel proposed that a unit factor that controls specific phenotypic trait, that is gene. Further, in 1902, Sir Archibald Garrod proposed that one gene is one metabolic block theory. In 1940, Biddle and Tatum said that one gene, one enzyme theory. In 1957, U.M. Ingram proposed about the one gene, one polypeptide theory. In 1960s, Yanofsky and co-worker said gene is a unit of recombination. And further, later in early 1970s, E.B. Lewis told that complementation test in Drosophila. So, the classical genetics considered a gene as the smallest unit of heredity, mutation, recombination, and function. So, work on microorganisms, especially bacteriophage. T4. Bacteriophage. What is bacteriophage? That is the virus which infects the bacteria. So the bacteriophage T4 revealed that a gene is divisible. This lead to the concept of the fine structure of the gene, where different functional units exist within a gene. Classical versus modern concept of gene. Classical concept: gene is indivisible, single unit for mutation, but fine structure of gene says that gene is divisible. Single unit for gene is single unit for mutation, recombination, and function. Fine structure concept says that separate units for mutation, recombination, and function. According to classical concept, abstract entity is there, but fine structure gene concept propose that there is a physical DNA segment. There is no internal structure according to the classical concept of gene, but fine structure of gene says that linear arrangement of nucleotides are the gene. Seymour Benzer's experiment 1955 to 1959, organism used bacteriophage T4 host Escherichia coli, gene studied ART2 gene, that is rapid lysis phenotype. Conclusion of Seymour Benzer's experiment, that is 1955 to 1959, on ART2 gene of T4 bacteriophage. The gene is not an indivisible unit, it has an internal fine structure. A single gene is composed of many smaller functional regions rather than being a single point. Mutations can occur at different sites within the same gene, not just between genes. Recombination can take place within a gene, proving that crossing over is possible at the intragenic level. The gene can be functionally defined as a cistron, unit of function, while mutant is the smallest unit of mutation. Recon is the smallest unit of recombination. So, the R2 region consists of two functional cistrons like R2A and R2B. Seymour Benzer demonstrated that genes can be mapped at a molecular level, and which is similar to chromosome mapping but with far higher resolution. A gene is practically a specific sequence of DNA containing genetic information required to make specific protein product. Prokaryotic gene is uninterrupted. It is a continuous structure, but in eukaryotic gene, the coding sequences or exons are separated by non-coding sequences which are called as introns. In complex eukaryotes, introns account for more than 10 times as much DNA as exons. The classical principles of genetics were deduced by Gregor Mendel in 1865 on the basis of breeding experiments with peas. He assumed that each trait is determined by a pair of inherited factors which are now called gene. Mendel's work was rediscovered in 19th century by Hugo de Vries, Courens and Zermatt. Wilhelm Johansen further coined the term gene in 1909, and in 1905 William Bettison coined the term genetics. Further, in 1972, Walter Fires and his team determined the sequence of a gene in bacteriophage MS2 coat protein. Richard J. Roberts and Philip Sharp found that the gene can be split into segments, making it possible that a single gene might be coding for several multiple proteins. Classical definition of gene. Gene is the functional unit of recombination and mutation, as one gene specifies one character. Modern definition of gene. Gene is the unit of genetic information, that is the sequence of DNA that specifies one polypeptide. This modern definition of gene includes coding as well as non-coding regulatory sequences. Recently, the Sequence Ontology Consortium reportedly called the gene a locatable region of genomic sequence corresponding to a unit of inheritance which is associated with regulatory regions, transcribed regions, and or other functional sequence regions. Now we go through the essential features or physical entity of the gene structure. It determines the physical as well as physiological character. We know that gene is situated in the chromosome. Gene occupies a specific position within a chromosome called as locus. Genes are arranged in a single linear order. They occur in functional states called alleles. Some have more than two alleles known as multiple alleles. Some may undergo sudden change in expression called as mutant gene or mutation, and it may be transferred to its homologous crossover or non-homologous counterpart, that is translocation. So it can duplicate themselves very accurately called replication, and it synthesizes a particular protein. So it determines the sequence of amino acid in the polypeptide chain, that is the genetic code. Gene is basically the functional unit of heredity we know. A gene is a segment of DNA that gives rise to a protein product or RNA. A gene may exist in alternative forms called alleles, but chromosomes in fact carry genes, and each chromosome consists of a linear array of genes basically in eukaryotes. So genes are classified based on their activity as housekeeping genes. They are constitutive genes that are required for the maintenance of basic cellular functions. They are expressed in all cells of an organism under normal conditions. Specific genes, they expresses for a specific type of protein when required by the cell. Prokaryotic gene structure, structural features, simple gene structure, small genomes 0.5 to 10 million base pair. Prokaryotic genes are collinear with their proteins. The image showing that this is a continuous sequence of nucleotides in DNA, and this is the continuous sequence of amino acids in a protein, and that is coded by this DNA. So DNA and protein is collinear in case of prokaryotes. Genes can be classified in various ways. The classification of genes is generally done on the basis of dominance, interaction, character controlled, effect on survival, location, genetic movement, nucleotide sequence of a gene, sex linkage, operon model, and ultimately their role in mutation. So classification and brief description of genes, that is a dominant allele or dominant genes that control, that expresses in the F1 generation, but recessive alleles or genes cannot whose effect is suppressed in the F1 and the recessive alleles are expressed in the F2 generation. Based on interaction, epistatic and hypostatic gene, when a gene that has masking effect on the other gene controlling the same trait, that is called as epistatic gene, or when a gene whose expression is masked by another gene governing the same trait is hypostatic gene. Major gene, a gene that governs qualitative trait. Such genes have distinct phenotypic effects. They are called as major gene. And when a gene which is involved in the expression of quantitative trait and the effect of such genes cannot be easily detected. Based on effect on survival, a gene which leads to death of its carrier when in homozygous condition, it may be dominant or recessive. Semi-lethal gene, a gene that causes mortality of more than 50% of its carriers, a gene that causes mortality or less than 50% of its carrier. They are subvital gene, and vital gene is a gene that does not have lethal effect on its carriers. Based on location, nuclear genes, genes that are found in nuclear genome in the chromosomes. Plasma genes that are found in the cytoplasm in mitochondria and chloroplast. They are also called cytoplasmic or extranuclear genes. Based on position, normal genes, genes that have a fixed position on the chromosomes. Most of the genes belong to this category. And jumping genes, which keep on changing their position on the chromosome of a gene. Such genes have been reported in maize. Based on nucleotide sequence, normal genes, genes having continuous sequence of nucleotides which code for a single polypeptide chain. Split gene, a gene having discontinuous sequence of nucleotides. Such genes have been reported in some eukaryotes. These intervening sequences do not code for amino acids. Pseudogenes, genes having defective nucleotides which are non-functional. These genes are defective copies of some normal genes. Based on sex linkage, sex linked genes, genes which are located on the X chromosome or sex chromosomes, they are called as sex linked gene. Sex limited gene, genes which expressed in only one sex or one gender, that is sex limited gene. Are sex influenced gene, genes whose expression depends on the sex of individual, say for example genes for badness in human. Based on operon model, regulator gene, a gene found in lac operon of E. coli which directs synthesis of a repressor protein. In lac operon, a gene which controls the function of structural genes, they are operator gene. Promoter gene, a gene in lac operon where RNA polymerase can bind, that is the promoter gene. And the structural genes, the genes which can synthesize protein through the formation of mRNA, they are called as structural genes. Mutable genes, genes which exhibit higher mutation rate than others, say for example which I genes in Drosophila, white eye gene in Drosophila, they are mutable genes. And mutated genes, genes which enhance the natural mutation rate of other genes in the same genome. Example dotted genes in maize, they are mutated genes. And anti-mutated gene, genes which decrease the frequency of natural mutation of other genes in the same genome. Such genes are found in bacteria and bacteriophages. So, genes practically have three regions. One is coding region, one is promoter, where the transcription start site is located at the junction of promoter and RNA coding sequence, and this is the site where RNA polymerase can bind, and ultimately there is a terminal region or terminator sequence. So coding region of gene, the DNA sequence that is copied into the RNA molecule or RNA transcript is the coding region. It starts with an initiator codon and ends with terminator codon. So there is no introns. So that is uninterrupted in nature, and it is collinear to its mRNA. That is the RNA coding sequence of a DNA. This is a promoter sequence and terminator sequence. After transcription, it will form a mRNA which have a 5' leader sequence and 3' trailer sequence, and in between the leader and trailer sequence there is a protein coding sequence which will participate in the process of translation and make the polypeptide chain, which is known as the ultimate product. Promoter elements, the upstream elements from the start of the coding region include promoter sequences. 50 to 100 nucleotides upstream of the start codon transcriptional initiation site or start site is there. So any nucleotide present on the left is denoted as minus symbol, and the region is called upstream element, such as minus 10, minus 20, minus 35, etc. And start site is symbolized by plus 1. Any sequence to the right of the start is downstream elements and numbered as plus 10, plus 35, etc. So this is the promoter elements of a gene. This is the start site. The left side of the start site there is a upstream element and they are designated with the negative symbols, and the right side is the downstream symbols and downstream element, and they are designated numbered as plus 10, plus 35, etc. So at minus 10 there is a sequence of Tata at or Pribnow box, and at minus 35 another consensus sequence Titigaka is there. So there are two there are most important promoter elements which can be recognized by the transcription factors. At minus 65 to minus 60 activator elements are there, and activation of the polymerase is performed. At minus 10 to minus 1000 enhancer sequence, it enhances transcription by 100 to 200 folds. So this is the enhancer, activator, Titigaka and Tata-at. Terminal region of the gene sequences for the termination of transcription. It takes place by row dependent mode or row independent mode. Eukaryotic gene structure. It has exons, introns, promoter sequences, terminator sequences, upstream sequences, downstream sequences, enhancer and silencer, upstream or downstream. So the signals include upstream sequence signal for addition of cap and downstream sequence signal for addition of poly A tail. So exons and introns, there is a difference. So exons are the coding sequence that are transcribed and translated. Introns are the non-coding sequence that separate the coding sequence. Exons code for amino acids in the polypeptide chain. Introns are any nucleotide sequence that are removed when the primary transcript is processed to give the mature RNA are called introns. Exons vary in number, sequence and length, but the introns, all introns share the base sequence GT in the 5' end and AG in the 3' end. A gene always starts with ends with exons. Some exons includes untranslated region or UTR, and introns were first discovered in 1977 independently by Philip Sharp and Richard Roberts. This is the image showing the alternative sequence of exon and introns. Promoter. A promoter is basically a regulatory region of a DNA located upstream expression. Core promoter is the transcription start site, that is at minus 34, which is the binding site for RNA polymerase, and general transcription factor also binds there. And proximal promoter, it contains primary regulatory element approximately minus 250 specific transcription factor binding sites are there. Tata box or Hogness box, that is minus 25 to minus 80, and CAT, that is upstream Tata, are two distinct sequences. These together are responsible for binding of RNA polymerase 2, which is responsible for the process of transcription. Upstream or 5' end. 5' UTR serve several functions including mRNA transport and initiation of translation, and signal for addition of cap, that is 7-methyl guanosine cap, to the 5' end of the mRNA. The cap facilitates the initiation of translation process, and it is also responsible for the stabilization of the mRNA molecule. Downstream or 3' end, 3' UTR or untranslated region serves to add mRNA stability and attachment site for poly A tail. The translation termination codon is TAA. TAA sequence is signal for addition of the poly A tail. So the terminator sequence is recognized by RNA polymerase as a signal to stop transcription. An enhancer region enhances the transcription of a gene up to few thousand base pair upstream. silencers, it reduce or shut off the expression of a nearby gene sequence. This is the eukaryotic gene structure and function. So what are the significance of introns? Introns don't specify the synthesis of proteins, cellular activities, but have other importance also. Many introns encode RNAs that are major regulators of gene expression. They contain regulatory sequences that control transcription and mRNA processing. Introns allow exons to be joined in different combinations, that is alternative splicing, resulting in the synthesis of different proteins from the same gene. Important role in evolution by facilitating recombination between exons of different genes are known as the exon shuffling. So exon shuffling has an important role in evolution that facilitate recombination between exons of different genes. This is the comparison of showing image showing comparison of eukaryotic and prokaryotic gene structure. In eukaryotes, there is upstream regulatory sequences. This is the open reading frame, which is open for the translation process, and further there is downstream regulatory sequence. But in case of prokaryotes, there is a regulatory sequences, and then there is operon. Some terms related to gene. So Benzer coined the new terms to denote the relationship between DNA molecule and genetic phenomenon. Recon, it is the smallest unit of DNA capable of undergoing crossing over and recombination. Muton is the smallest unit of DNA which can undergo mutation. Cistron is the unit of function. It is the gene in real sense capable of synthesizing a polypeptide chain of an enzyme. Complon, it is the unit of complementation. Marked ends of polynucleotide chain. Each polynucleotide chain has marked ends. Its top end has a sugar residue with free 5' carbon atom which is not linked to another nucleotide. So the triphosphate is still attached to it. This end is called 5' end and 5' phosphate termination. The other end of the chain ends in sugar residue with C3 carbon atom, that is not linked, and it bears 3' OH group. This ends of polynucleotide chain is called 3' end or 3' OH termination. It means that the polynucleotide chains have direction and they are marked as 3' to 5'. This is the image showing the transcription and translation process from the DNA. There is a coding strand and a template strand. The template strand is responsible, it's complementary to the newly synthesized RNA, and coding strand is the same as the amino acid chain, which is produce the polypeptide chain after translation. So materials required for the synthesis of RNA, that is the enzyme and RNA polymerase, DNA template, all 4 types of ribonucleotide triphosphate such as ATP, CTP, GTP and UTP, bivalent metal ions such as Mg2 plus or Mn2 plus as a cofactor. So this is the double-stranded DNA and RNA, that is coding or sense strand, and template or antisense strand. The original transcript formed from DNA is called heterogeneous nuclear RNA or hnRNA. hnRNA basically contains sequences of both introns and exons. The introns are removed from hnRNA by a process known as splicing. After splicing, the RNA is converted into messenger RNA or mRNA. So the regulation in gene action at post-transcription level, that is in eukaryotes, there is splicing. So splicing always removes the intron, non-coding regions that are transcribed into RNA, so in order to make the mRNA able to create proteins. So cells do this process by spliceosomes, that is composed of small nuclear ribonucleoproteins and snRNPs binding on either side of an intron, looping the intron into a cycle and then cleaving it off. There is the 2 ends of the exons are then joined together. This is the showing of exon, and this is the exon in intron boundary are there, and this is the process of where from the pre-mRNA mature mRNA is produced. So Philip Allen Sharp is an American geneticist and molecular biologist who co-discovered RNA splicing, and he shared the Nobel Prize with medicine in physiology in medicine in 1993 with Richard J. Roberts for the discovery that genes in eukaryotes are not contagious strings but contain intrions, and that the splicing of messenger RNA to delete those introns can occur in different ways, yielding different proteins from the same DNA sequence. So he has been selected to receive the 2015 Othman Gold Medal also. So discovery of introns in eukaryotic DNA and the mechanism of gene splicing. So what is the introns? Introns are any nucleotide sequence within a gene which is represented in the primary transcript of the gene but not present in the final processed form. The overall length of a gene is determined largely by its introns. Introns range in size about 50 nucleotides to more than 1 lakh nucleotides. Exons are usually short. Typically, they are present on the order of 150 nucleotides, but introns usually do not code for proteins. However, certain introns of group 1 and group 2 classes contain open reading frames that are translated into proteins. So, this is the image showing the prokaryotic mRNA, and this is the eukaryotic mRNA. This is the average size of exon and introns in human genes. This is a gene product, size of gene, number of exons, average size of intron. There are types of most common introns. There are GUAG introns, where it is found in eukaryotic nuclear pre-mRNA. AUAC introns are found in eukaryotic nuclear pre-mRNA. Group 1 introns are found in eukaryotic nuclear pre-rRNA, organelle RNAs, some prokaryotic RNAs, and group 2 introns were they found in organelle RNAs, some prokaryotic RNAs. Pre-tRNA introns are found in the eukaryotic nuclear pre-tRNA. So there is archaeal introns, which is found in various RNA. These are adapted from T.A. Brown, genome 3. Both of it is also adapted from T.A. Brown. So we must know the functional sequences in promoter region in prokaryotes to know ultimately the fine structure of the gene. The promoter region in prokaryotes, there are 2 or 3 important functional sequences which are called as consensus sequence. These sequences from promoters of different cistrons differ only by 1 or 2 bases. So there is Pribnow box 10 region, a consensus sequence in 35 region, and upstream promoter element is there. So the Pribnow box sequence orients RNA polymerase on promoter and helps it in locating the precise bases at which transcription will begin. A consensus sequence in -35 region, this hexanucleotide sequence is recognized by sigma factor of RNA polymerase. It is presumed that sigma subunit of RNA polymerase binds to -35 sequence, and upstream promoter element, AT-rich recognition element in the promoter region, which is present in the promoters of certain highly expressed genes and is located between 40 and 60 nucleotides. This sequence is bound by the alpha subunit of the RNA polymerase. So the image showing the functional sequences in promoter region found in prokaryotes. So the functional sequence in promoter region in eukaryotes, that is the sequences of nitrogenous bases similar to that found in prokaryotes, are present in the promoter region of eukaryotes also. So there is Tata box or Goldberg-Huggins box, the hexanucleotide sequence consisting of 5' Tata TATAA 3' is usually represented by TATAX, where X may be TOR A. It is the site of assembly of pre-initiation complex, that is the transcription factors and polymerase, which is essential for initiation of transcription. So in addition, eukaryotic genome has proximal control elements named as GC box with base sequence GGG, CGG at -10 site, and cat box with base sequence GCCC TCT. So the image showing the eukaryotic gene structure, here is the proximal control elements, and then there is core promoter, there is and the open reading frame where there is start of transcription, there is poly A tail, and the open reading frame is interrupted by there are introns and exons are there, and just two sides of the open reading frame there is a 5' untranslated region or leader sequence and 3' untranslated region or trailer sequence. So as we conclude, remember that the fine structure of the gene reveals a gene as a precisely organized DNA segment, not a single indivisible unit. But understanding Recon, Muton and Cistron, we gain clarity on how recombination, mutation and function occur within the same gene. This concept depends on insight into genetic variation, and gene mapping, making it essential for undergraduate, postgraduate, and NET preparation. So keep revising, think conceptually, and continuous exploring the elegant details hidden within the genome. Thank you.